help me, Omri Amirav-Drory, to stage. Thank you.
So I thought that I was speaking fast, but after the last one, Moran is speaking really fast. So I'm really happy to be here and to speak here, especially because the name of the conference, so DLD stands for Digital Life Design. So I thought it's a synthetic biology conference because that's what we do. We design living things using digital tools. So again, we're in the space of synthetic biology, and we love living things. So biology is technology. Biology is the most advanced technology on Earth. These little things, so we talk about Moore's law, where we double the transistor number every 2 years. Those little things, they double every 20 minutes. So biology is very powerful. It's scalable, and it can solve some huge problems we have in the world. So for us, a big motivation for our company is just a realization that we live in a civilization totally dependent on finite resources of oil, coal, and natural gas to produce everything. So not just energy, but also chemical, pesticides, pharmaceutical, almost everything we use today come from finite resources. We should look at nature. Nature has been around for at least 4 billion years, mainly because it can use renewable resources such as sunlight and sugar and CO2 to produce everything we get from fossil fuels. So how do you code things in biology? So all of you, I guess, have cell phones. Cell phones are computers. What computers understand is this kind of code, binary code, 1 and 0. But nobody really codes today by typing 0, 1, 1, 0. They use abstraction layers. They use tools to design, debug, and compile their code into different applications running on computers. Well, biology is very similar. Instead of coding in 1 and 0, you code with genetic code, with A, T, G, and C. The applications are amazing and can really change our civilization. They run on this kind of hardware. So it's not hardware or software. We call it wetware. So biological computers, just cells. But what we don't have in biology is the same tool to design, debug, and compile biological code to create a bio application faster. And that's what we are building. We are trying to just accelerate the design, build, test, learn cycle in biology. Help research scientists build bioapplication faster and better. There are already some very cool applications of synthetic biology in the market. The company on the left, Emrys, they built, their first application was an anti-malaria drug called artemisinin. This drug is usually being produced by a plant. They took the genetic code from the plant that codes for the enzyme that create this molecule and moved it into yeast. And in just 1 year of production, they produced 50 times the world production and saved 160,000 people. Just 1 year, one drug, one application. Another application is ability to create vaccine much faster. And the same company here, Synthetic Genomics, that create vaccine, also just last year, very fitting to Israel, got a new project to create humanized pigs. It's not what you think. It's genetically modified pigs that you can use their organs for transplantation without causing any immune response. So think about that, organs without limitation. Solozyme is another company, started as biofuel, but now they are also producing cacao butter, the main ingredient of chocolate. So instead of burning forests in South America to grow cacao beans, now they do it in algae. So just fermenting cacao butter. And you can also produce spices like vanilla extract and other things. So if one creature can make it, another creature can do it also. So why does it happen now? The main reason synthetic biology exists is because of two technologies that are moving super exponentially. So we all know about Moore's law, right? Computer getting faster, better, and cheaper. In biology, we have two Moore's law. One of them is reading DNA, so DNA sequencing. I guess you all heard about the Human Genome Project, right? It was about 12 years ago. It cost $3 billion to sequence one human genome. Today, you can do it in less than $1,000, right? It's one of the technologies that moved the fastest ever in human history. So more and more, we're digitizing biology. We're taking all the code that nature created, whole ecosystem, and turning it into ones and zeros to code in the computer. But the real cool technology is not reading DNA, it's writing DNA, because now you can create any code you want from scratch. And the price of writing DNA has also been dropping down very rapidly. It's still about 6 to 8 orders of magnitude more expensive than reading DNA, but it's going down, and it's going down fast. And it's not just the price of writing the code of life is getting cheaper. Our capability of stringing together larger and larger designs is getting exponentially higher. So in the beginning of the 2000, the largest code we could create is about 1,000 nucleotide, 1,000 letter of code. And then at around 2005, we were able to create the first genomes, and that was viruses. Viruses have a very small genome. So, for example, the HIV virus, just 9,000 letter of code, or Ebola, just 18,000 letter of codes. And then in 2010, Craig Venter and scientists from the Craig Venter Institute, for the first time, synthesized a whole organism. So it was 1 million base pair, the entire code for a self-replicating bacteria. And they even booted it up. And because they wrote the code from scratch, they even wrote their name into the code and even an email address. So there is a bacteria now, lives in the lab, that if you sequence it, you can see an email address and send emails to the researchers, which is pretty cool. And then just a few months ago, researchers have synthesized a yeast chromosome. So yeast have a chromosome just like us, just smaller. So for the first time, synthesizing a whole eukaryotic chromosome, and again, the capability are just growing. And this is where we build our software, to handle the complexity of forward engineering in biology. So we build a complicated software where you can load entire genomes, do metabolic engineering, check that nobody's trying to order Ebola, for example, which is possible, and somebody actually did try to, several people. Debugging rules, design rules. And the vision is that if you go to Rothschild Avenue and you see all these coders working in coffee shop, maybe in the future, instead of coding the next social mobile app, they'll be coding a living thing to solve some big challenge we have in the world. So if they have an idea, using our software, they are connected to huge bioinformatics databases and to biofabs. Those are companies that you can send your sequence, they make the DNA and then test it to create products. And that's how we make money from the software and from a marketplace for bits and atoms. But what we really like about this space is that it's being democratized. The price is falling down, the technology is getting in the hand of everyone. So there is a new movement called DIY bio, do-it-yourself biology, so the maker movement coming to biology. The guy on the right is the first guy in Europe that got permission to do genetic engineering in his kitchen, so you can actually see the pots and pans. So genetic engineering is very much like cooking. There are even community labs, so GenSpace in New York and BioCurious in Sunnyvale, great name, BioCurious, and I'm definitely trying to start one in Israel because I think it's crucial. Those are places that for $100 a month you can get access to lab facility. So you don't need a lab, you just need to have an idea. There is even a competition for high school kids and undergrads to create the best genetically modified organism. It's called the iGEM competition, the International Genetic Engineering Machine Competition. It started at MIT in 2004. Now it's all over the world, including amazing groups from the Ben-Gurion University and the Technion. And let me show you what some undergrads can do over the summer break. So this is a group from Munich a few years back. So Munich, Oktoberfest beer. So of course, you know what makes beer, it's yeast. So they thought, let's modify yeast that instead of just making alcohol, which is a boring molecule, we create some other things. For example, an anti-cancer drug, or the molecule that make lemon taste like lemon, or a protein, which is a natural sweetener, or even caffeine. And they brought to the competition a beer bottle that was naturally caffeinated because the yeast that created it also create caffeine on the way. This team did something amazing. They re-engineered a gut bacterium called E. coli to produce different pigments, different colors, corresponding to different disease states. And the idea is that in the future, you will just eat a probiotic yogurt, and when you go to the bathroom, it's quite graphical here, depending on the color, you can see what you have. So, oh, it's green. Wow, I need to go to the hospital. Which sound like funny, but it's also very cheap and accessible diagnostic. This group from Cambridge took the bioluminescence system from marine bacteria and improved it so much they can actually read on the top left side the Jungle Book in the light of glowing bacteria. So when we saw this project, we thought to ourselves, let's show what's possible today. Let's show what people can do using our platform. So we decided to make other projects. So we decided to go to Kickstarter and show people that you can crowdfund biotechnology. So we set up to do a project to create glowing plants that contain the same machinery of fireflies and they can glow. And this is the video from the Kickstarter campaign.
Problem with plants, they don't glow. Guess what? We're fixing that.
The main problem with plants, they don't glow. We asked for $65,000. We got $500,000 for almost 9,000 different backers. And the company, now it's spun off to a company that just a few months ago got into Y Combinator as one of the first wave of biotechnology companies ever in Y Combinator. And I want to show you something. The next picture was taken a few months ago by the Wall Street Journal. This is a real plant. Can you see the guy's face? So it's actually working, and I came back from the US 2 days ago and actually carried with me some seeds of the plant, so they are here with me, and I'm about to, no, you're not getting it now, so I'm about to grow some of them in the Holy Land, so if you want some glowing plants, I have them in my pocket. So we did this project, and then my team asked me, what next, right? That was cool. So I thought back to this project, this iGEM project from MIT from 2006. So what they created is a gut bacterium that when it grows, it smells like mint. And when it stops growing, it smells like banana because it expresses different proteins that produce different chemicals. So we thought, what kind of product can we make with this? So we made this video. And you'll be one of the first people to see it, so let me know what you think about it. Would you use this product?
We've all been there. Nature calls right when you don't want it to. You know the problem. Dropping your atomic stench at the wrong place at the wrong time with nowhere to hide and only inadequate ways to cover it up. Tired of messy air fresheners that just try to mask your odor with chemicals, awkward solutions that just admit guilt, or being left in the stench with no solution at all and having it ruin your day? Don't you wish you could cut that ugliness at the source? Well, you can. Introducing Petomics, a revolutionary new dietary supplement.
Okay, sorry about that. Let's try it again. I think you got the gist of it, though. One second.
With chemicals, awkward solutions that just...
Okay. If it doesn't work this time, I think I'll just tell you what's going to happen. I think it's pretty obvious.
With chemicals, awkward solutions that just admit guilt or being left in the stench with no guilt or being...
Wow. This is Internet Explorer. That's what happened. So I guess you can imagine what happened. Genetically modified bacterium, that probiotic that you can just eat and change things for the best, we think. And that was the seed of a new company called Personalized Probiotics, now working in San Francisco. We are covered in bacteria on our skin and in our gut, and if we can modify it, we can do a lot of very cool, interesting things. But it's not just about making your fart smell like mint, which is cool, but not really important. We live in this biological living world. The air we breathe came from biology. We have no future if we don't use these tools of synthetic biology to build a more sustainable future. And if high school kids, undergrads can do it during the summer break, so can you. So join this revolution, and thank you very much.
Amazing. So I'm going to explain what is going to happen now. I'm going to call on stage